Thank you, everyone. I'm going to present, my name is Maria Juliana Pardo. I'm a master's student supervised by Etienne Liberté, and I'm going to present my project called Optimal Detection Time of Phragmites Australis over a growing season. Human-induced climate change and involvement in natural habitats is altering plant communities worldwide and facilitating biological invasions by intentional and accidental movement of species. Invasions can alter biodiversity and ecosystem functioning with potential negative consequences on ecosystem services as well as on agricultural and forest resources. As well, they represent a large economic cost for societies. As an example, as of the year 2005, the cost of invasive species management in the United States reached $120 billion per year. An invasive species is defined as a non-native that spreads rapidly and can have negative ecological, environmental, and economic effects on environments where it has been introduced. Invasions have complex and usually long-term direct and indirect impacts that often become apparent only after invaders are well established and have acquired large ranges. To this date, most invasive species management has been reactive, but the future of the field is in prevention. Being able to predict when invasions would occur and at what pace, ideally could lead us to prevent them. Among the list of Canada's most invasive species, we found the common reed Phragmites Australis colonizing wetland ecosystems. Phragmites Australis is a cosmopolitan perennial grass that grows very tall, forming large and dense colonies. Research into its ecology has revealed that it exhibits many characteristics of a successful invader, like the combination of sexual and vegetative reproduction, high biomass production and retention, and root-derived allelopathy that inhibits the growth of neighboring competing plant species. The establishment and rapid spread of Phragmites is a threat for biodiversity of wetland ecosystems because it transforms diverse communities into low diversity common reed stands. This has a major effect, altering ecological processes such as nutrient cycling and flooding regimes that in turn feedback negatively to the functioning of wetlands and affects associated fauna. For this reason, the spread for the common reed is a major conservation challenge, and its management and eradication are a conservation priority. In North America, invasive and native lineages of Phragmites can be found. The invasive haplotype M subspecies Australis was introduced in the United States and southern Canada in the 19th century. In the province of Québec, was considered a minor component of wetland communities, with its distribution, being restricted to the shores of the St. Lawrence River. With the development of the highway network in the 1960s and 1970s, it expanded its range inland and initiated a large-scale invasion that continues to this day. The extent of this invasion is projected to accelerate due to the effects of climate change. Rising temperature and altered flood regimes that are being experienced in the hydrographic region of the St. Lawrence and the Great Lakes causes low water levels and warmer temperatures during summer, creating suitable habitats for the establishment of the common reed. Today, genetic analysis estimate that more than 95% of colonies of Phragmites are dominated by the invasive haplotype M. A recent study using high-definition satellite images and a combination of multispectral bands mapped the current distribution of Phragmites along the St. Lawrence River. The study identified existing common reed stands and modeled the spatial distribution of uninvaded but suitable germination grounds for these species. In red, you can see them there. The study reports that stands of small size remain undetected and a substantial increase of suitable area for invasion. The spatial model accurately classified large stands of common reed, but it could not predict when an invasion will occur or the speed at which it will progress. Lastly, they predict a staggering loss of biodiversity in the St. Lawrence River wetlands as the common reed continues to expand. These results underscore the importance of developing tools for detection in early stages of invasion for the preservation of these ecosystems. Spectral data, which describes the interaction of plant leaves with sunlight, is a remarkable tool for species discrimination and is increasingly being used to map invasive species. Species discrimination can be achieved by evaluating differences on spectral signatures. In principle, sufficient high spectral resolution should permit unique discrimination of individual species based on how dissimilar their spectral signatures are. Regardless of all plants being spectrally similar, it is known that more functionally different and more distantly related species are more spectrally dissimilar. With regard to invasive species, this presents an advantage for their detection, given that most often they possess traits that are novel to the colonized environment, and therefore should be more easily discriminated. Extended leaf phenology, that is earlier leaf emergence in spring and or prolonged leaf retention in autumn, has been proposed as a key feature driving events of invasion in northern latitude deciduous ecosystems. Irrespective of whether this mechanism represents an advantage to invasive species in terms of use of resources, or whether they are more responsive than natives to warming trends that have lengthened the duration of the growing season, accounting for phenological patterns provides an opportunity for spectral detection of invaders. Beyond discrimination of a target species, detection in early stages of invasion is lacking and is primordial to prevent adverse effects on ecosystem functioning and biodiversity. There are many traits that are strongly tied to phenological state, and their change in direction and magnitude over the course of a growing season can be captured in their spectral signatures. Leaf scale biochemical and anatomical traits that change through time, like leaf pigmentation, nutrient concentrations, and leaf mass per area, are described by specific wavelengths of the electromagnetic spectrum and therefore influence the spectral reflectance behavior of leaves. Into temperate deciduous forests in the United States, the visible and the near-infrared regions of the spectrum changed in amplitude and shape through one growing season, while the shortwave infrared region remained more stable. This is consistent with a study in the Peruvian Amazon that reported that the most age-sensitive spectral domains are the green peak, the red edge, the near-infrared region, and the water absorption features around these wavelengths. Forgot my numbers, sorry. Time series data that describe seasonal changes in leaf traits and spectra show that there are critical time windows that capture the maximum differentiation amongst co-occurring species. This knowledge is critical to improve spectral differentiation models that target invasive species. Building on previous work in our lab that has shown that Phragmites might be confused with co-occurring species like the reed canary grass, we used a time series of leaf and spectral data collected in the islands of Boucherville in Québec to evaluate when is Phragmites more distinct from co-occurring species and to determine the optimal time for its early detection. During the growing season of 2019, we studied 5 different species: Bromus inermis, Phalaris arundinacea, Phragmites australis, and the genus Solidago antifa. 10 samples per species were collected every 3 weeks from June to September and were analyzed for spectral and functional trait measurements following Cabo protocols. This project seeks to understand and answer 4 questions. Can invasive species be discriminated from others using leaf spectra? Does species discrimination vary during a growing season? Do foliar traits change over a growing season? And are these functional differences among species captured in their spectra? Our hypothesis is that Phragmites would be more different from its neighboring species towards the beginning or the end of the growing season due to its extended leaf phenology. During the middle of the growing season, resource acquisition strategies of all species would be more similar, and therefore species spectral separability would be less accurate. I will share some preliminary results with you. Analysis of coefficient of variance in the spectrum show us that in general, the coefficient of variation across all species were lower for Tifa, Phragmites, and Phalaris. For only Phragmites, the coefficient of variation in the near infrared region was significantly lowest in the second wind, in the second week, and in the shortwave infrared region was also significantly lowest during the first and the second weeks of sampling. Second, our PLSDA model allows us to discriminate Phragmites and the other species with high accuracy. It also shows us that the accuracy of discrimination does change throughout the season, and our results indicate that Phragmites spectral separability is maximum in the early stages of the growing season during weeks 1 and 2, and then again at the end of the growing season in September during week 6. We also explored the temporal dynamics of leaf traits and found important patterns. The photochemical reflectance index, indicative of light use efficiency, showed an increase at the beginning of the season, reaching a peak around mid-season corresponding to leaves being mature, and a subsequent decline towards the end of the season, with photosynthesis declining when leaves approach senescence. The same pattern is evident in the amount of chlorophyll through time, where we can see a peak towards the mid-season and then a decline that corresponds to chlorophyll breakdown as leaves start to senesce. Increased leaf mass per area through time has been associated with light harvesting and with an increase in total carbohydrates and lignin, leaf thickness, and density. Leaf water content and leaf mass per area usually exhibit a negative correlation amongst them, and leaf water content has been indicated to be the most important leaf trait for modeling leaf age. In our results, we found that leaf water content showed a decreasing trend with time across all species. Lastly, the CN ratio captures an important trend of nutrient acquisition, with nitrogen being allocated to leaves earlier in the season and carbon accumulating faster when leaves are more mature. This results in an increased CN ratio throughout the season. The answer to the fourth question of whether species functional differences are captured in their spectra is to be completed. Thank you.
Thank you very much, Maria. So if you have questions, so no one has raised their hand. And I don't see any question. Mark has a question from Mark.
Yeah, just got to get myself unmuted here. Yeah, I'm curious. So you can discriminate species based on the spectra. Do they discriminate in functional trait space? Like if you were just to, do you know whether or not if you just used functional traits, does Phragmites really stand out in the functional trait space too?
Well, their functional traits do change. There are certain patterns, but whether that is captured in the spectra, I haven't got to that analysis yet with the PLSR regressions.
But do the functional traits of Phragmites, are they really distinct from those other species that it co-occurs with, or aside from whether they change through time? Are there or do they overlap a lot?
Not as I expected. And I think what I've read in the literature, Tifa is a very, Phalaris gets confused with it a lot, but in respect to functional traits, Tifa is also Tifa tends to be the more dissimilar one, although it's not an invasive species. So what I was expecting is to find a very, like a big distance in functional traits from Phragmites to the rest of the group, but Tifa kind of breaks that relationship a little bit, and then Phragmites does not seem to be that different in functional traits.